Hello, back from the break. We'll start the next session titled Multi-omics Data Integration Based on Artificial Intelligence and Big Data. And I will present briefly my colleague, Xavier Domingo. He is the scientific coordinator of the GLOMICAVE and PhD researcher of the Omics Sciences Unit. He holds a electronic engineer and MS and PhD in bioengineering. He carried the postdoctoral at the Scripps Research Center for Metabolomics, and then later moved as principal investigator at Eurecat within heading the bioinformatics group's medicines lab at Eurecat within the Omics Sciences. He has been also as a professor at the Department of Electronics at the University of Rivera Virgili, and he has been the PI of research projects also at national and European level. So, Xavi.
Thank you very much, Biotza. Thank you for the presentation. So I will introduce you a little bit the platform, the GLOMICAVE platform, in a different way, at least explaining what how the different modules of the platform are joined together and how are they interconnected. So I will start by providing an overview of the platform, and then I will explain how the different modules from extracting data from omics data sets and from literature are connected. And finally, we'll finish with the how the multi-omics integration algorithm works. Now, so the GLOMICAVE platform is a platform, so it's a web application that you can access and allows you to to upload the data into the platform, to integrate it. So you can upload the multi-omics data, you can upload metabolomics, transcriptomics, genomics, proteomics data, and integrate it and contextualize it also into pathways. And you can make use of unique knowledge base that we have created using data and text mining analysis. So we have extracted the data. We have done data mining over public omics data sets, and we have also mined scientific text, scientific literature, and processed it, as Raman just explained you a moment ago. We have analyzed that data and create a knowledge base that has relationships between genes, proteins, metabolites, traits, and so on. And you can use that knowledge base to contextualize your results also with. You can visualize the results in an interactive and easy way fashion, as I will show you. And you can also query this knowledge base independently. You will see all this part in a moment. So but I would like to start by explaining how the different modules work, or how an overview of the different modules. First, I will go through the data mining module. To do this, to extract information from the public repositories, what we have done is use data mining approaches over public databases, including metabolites, Geo, ENA, RAExpress. And in this database, we have two types of data. The first type of data is the annotated data, the data that has been already processed, and you have the name of the genes or the transcripts, and then you have the concentration in the columns, let's say. This data is very easy to integrate, so we have already integrated. It's very, let's say, you don't have to do any pre-processing. You can integrate and process it for further analysis. But then on the other hand, we have the unprocessed data, the raw data. And this data is a little bit more challenging to process, of course, because it's a computational burden to process all this data, but we have managed to process some of the data using also a Galaxy workflow and being able to transform that raw data into annotated data to integrate that into the knowledge base. But another additional problem is that comparing or using data across data sets from different experiments is also quite challenging. So we have managed to pre-process transcriptomics data in a way that we can compare this data across across different experiments. And interestingly, just recently, NIH has released a similar thing, a matrix of expression data where multiple experiments can be compared. So after so many years and considering all the NIH budget that the NIH has, they have released what I consider the GLOMICAVE's counterpart for non-human expression data. So in fact, we have been able to create something similar to this, but for non-human research. So this is the data mining part. Let me explain you a little bit, or give you a glimpse of the how have we integrated what Roman has you explained a moment ago into the platform, extracting information from literature. When it comes to extracting information from literature, I consider that there is like two parts, right? The first is what I consider to be the explicit information that you can extract from the literature. And if you consider this example of a sentence or a title of a paper, where this photophilic B1 and vernalization A1 genes are associated with altered flowering timing width, it's, let's say, at least for a human, very intuitive to extract information or to condense or to structure this information into that these two genes are related to this trait, to this phenotype, right? For a computer, is much more challenging, as Roman has you explained a moment ago, but this part is what, let's say, anybody could manually do, right? Extract explicit information. And well, we can create, as Roman showed you a moment ago, we can create a large network of connections, how the different traits, how the different proteins, how the different metabolites are connected based on this information that we can extract automatically from literature. And however, we provide tools within the platform to query this database, to query this large knowledge graph in an easy way. And so let me show you an example of the platform, actually, how you can query the platform, how you can query this knowledge graph within the platform. So you can, this is the GLOMICAVE platform, where the different modules are here in the top row, and you can click and access to different parts. And if you click on search, you can search this knowledge graph independently, which is the first, what I said before, that you can query this knowledge base, introducing any multi-omics data or introducing any omics data of your own. And for example, if you do a search like threonine and serine, and you select those are small molecules, and you click here on finding publications, you will be able to, it will provide you a list of the papers where threonine and serine are co-mentioned, also the related terms, and then will give you the papers where threonine and serine are mentioned alone. So if you click on details, you will see here the different papers. It will list you different papers where the serine and threonine are mentioned. And as you can see, it will highlight where it mentions those terms in the text. And for example, here you can also see some genes. In fact, it actually works with genes. So if you add ATR1 gene in the query, it will also come will show you the papers that have this gene mentioned also with serine and threonine, or all the other terms that you introduce in the platform. So in fact, it also works with synonyms. If you use lactic acid as a query term, the lactic acid can also be mentioned, it's known also as lactate, without entering into a chemistry discussion. And you can, and in fact, our approach is also, as Roman showed you a moment ago, is able to recognize these synonyms and highlight these terms within the abstract. So as I said a moment ago, we have explicit information, but on the other hand, we also have the implicit information. And what is the implicit information? It's the one that you don't see, that the human could not have recognized, even if you give it this information, even if you give this information to a trained human. And how does it work? Let me just give you a glimpse of how this approach is implemented into the GLOMICAVE platform. So imagine that we have one paper where one gene A is co-mentioned with metabolite A and B, and another gene, another protein. And then we have another paper where this same gene A is co-mentioned with metabolite C and gene C. And not just two papers, imagine a lot of papers. Well, by using advanced machine learning techniques, what we can do is let's say, consider that if this gene is combination with this and this gene is combination with these other genes, it makes sense that these are also related, that these genes, or sorry, these metabolites are also related among them, among each other, right? If gene A is related to this and gene A is related to this, it makes sense that this is related to this and this. This is a very simple explanation of how the algorithm works, but in fact, it's much more complicated. And the extraction of this implicit information has been previously used to discover different relationships that otherwise would have remained undiscovered, right? That's like these ones that I'm showing here on the screen. And in the GLOMICAVE platform, we have been able to extract relationships among entities. For example, proteins. We have been able to extract protein-protein relationships that we have compared with known knowledge, by comparing these relationships with a known database of protein-protein relationships, like string database. And we have been able to discover new associations that were not reported in this database. So as I showing you, we can use these data mining techniques and these natural language processing techniques to extract information from omics data and public repositories using machine learning and other techniques. And as Pierre showed you also a moment ago, we have integrated into the platform an auto machine learning module to enable a wide base of scientists without any knowledge about machine learning, or minimal, let's say, knowledge about machine learning, to use the machine learning in an easy way in their experiments. So the idea is that you can upload your data, your multi-omics data, and the AutoML module is going to be able to extract the relationships among the entities, among the proteins, and so on, in an easy, let's say, in an intuitive fashion without any knowledge of how to code the actual algorithms, right? So, of course, the GLOMICAVE platform aims at integrating different omics technologies, different omics data, including metabolites, including metabolomics data. But the issue is that metabolomics is not as mature as the other omics technologies. While with genomics, transcriptomics, proteomics, you can identify hundreds or thousands of genes, proteins, or transcripts. With metabolomics, you can only identify a few of them, right? And so one of the aims of GLOMICAVE was also to try to put metabolomics a little bit on the same level as the other omics sciences. So to that end, we have also developed an algorithm that is capable of annotating in a very easy and intuitive fashion the metabolites that you observe in untargeted metabolomics data. So being able to translate this untargeted metabolomics data, where it's very difficult to identify and annotate metabolites, in order to contextualize the possible, to extract what possible metabolites are being observed in your data, and also, as I said, contextualize these possible identities within the network, within the biochemical pathways that you observe in your data. So I just explained how you can query a knowledge base. I have explained how this knowledge base has been created. Let me show you a little bit how you can upload the data, how you can integrate the data, and what do you feel, what are the results of the GLOMICAVE platform. So as I said, the platform is pretty intuitive, so you can only have to drag and drop the data into the platform, and then you can create a new project with this data. So you drag your data, your multi-omics experiment, you create a new project with that data, and then at the end, you get many other things, but one of the main things that you get is a list of these regulated pathways in your data. So you can get a list of these pathways, and then if you click on any of these pathways, you will see the specific information about the pathways, the specific genes, enzymes in this case, and metabolites, and it will highlight which elements are dysregulated in your data. It also does the statistical analysis for you, and so you can go through this data. You can also contextualize or integrate the information from the AutoML module. You can also integrate the information from literature, as I showed you a moment ago, so that the user can easily integrate the multi-omics data and get a detailed explanation of the results from the data from their experiments. And as I just show you some screenshots and glimpses of the front end of what you actually see in the web application. But this web application is actually composed of two things: what you see and what you don't see, which is the back end and in computational or informatic terminology, which has also been quite challenging to create, because of course creating a database, or sorry, a platform that is able to support this large volume of data and is able to process and analyze all this kind of data is quite challenging. And you will see more details in the upcoming explanation by our colleagues that have developed this backend strategy. So that's all on my side. Thank you very much, and I'll be happy to answer any questions that you may have.
Presentations, and then we will collect all and then we'll be posting the questions to each of you. So very connected now with previous Xavier presentations. We will follow with our next consortium partner, Rodrigo Caballero. He is biomedical engineer from the Polytechnic University of Valencia, with more than 4 years experience in big data solutions, mostly focused on the data cleaning and storage. He's currently working in disciplines more related to cloud technologies and data exploitation, and he is data engineer at Tree Technology. So please, Rodrigo.
You can see my screen already. Okay, perfect. So as Xavier has told, I'm going to talk about the architecture and infrastructure that we have developed for this project. So the main points that we are going to achieve in this presentation is the architecture, the application, the different parts that the main parts that the application have, and also the concept of data lake and databases and the difference that they have. Now, first of all, the architecture that we have choose for this project is a cloud architect, and we are using Amazon Web Service to guarantee the advantages of cloud architecture, which would be the remote access that enables remote access to resources, allowing teams to collaborate efficiently from everywhere in the world. This flexibility promotes productivity and enhances collaboration among distributed teams, whether working from the office, home, or on the go. Team members can access the same resources seamlessly, which was key for this project that we have different teams from all around Europe. The scalability is another very important part of the cloud architecture, because the cloud provides offer on-demand scalability, allowing users to easily scale resources up or down based on the demand. This elasticity ensures that applications can handle fluctuations in traffic and workload without experiencing performance degradation. As demand increases, additional resources can be provisioned instantly, ensuring a seamless user experience, which was key to have a development environment and also now a production environment where the amount of data that we are dealing with can change. And another key point to use the architecture, the cloud architecture, would be the pay-per-use. The cloud computing follows a pay-per-use pricing model where users only pay for the resources they consume. This cost-effective pricing model eliminates the need for large upfront investments in hardware infrastructure. Instead, users can scale resources as needed and pay only for what they use, optimizing cost efficiency and resources utilization. That this is something that we are gonna enjoy now, because when the development environment is not anymore needed, we can just switch off and use the resources that we need for the production environment. The main key or the main services that we have used from the Amazon Web Services, it would be the Amazon SageMaker. That is a fully managed machine learning services that enables developers and data scientists to build, train, and deploy machine learning models at scale. With SageMaker, users can streamline the entire machine learning workflow, from data preparation and model training to deployment and monitoring. The service offers built-in algorithms, managed notebooks, and automatic model tuning, making it easier to develop high-quality machine learning models quickly. The next service it will be the Elastic Container Registry. Amazon Elastic Container Registry is a fully managed Docker container registry service that makes it easy to store, manage, and deploy Docker container images. The ECR integrates seamlessly with other Amazon Web services, allowing users to deploy containerized applications on Amazon Web Service infrastructure. The service offers features such as encryption, access control, and image lifecycle management, ensuring secure and reliable container deployment. This is a key piece in our infrastructure because we can have separated services, for example, the front-end resources from the different models that the different team can allocate in these different images, so we don't compromise the development of one of the teams with the development of the others. And also, this is an important piece for the continuous integration framework because we can save the images that we deploy in the past. Even if we want to reset this container, these services allow us to do it so. The next service that we have used is the Amazon Web Service Neptune. That is a fully managed graph database services that enables users to build and run applications that work with highly connected datasets. Neptune supports popular graph query languages such as Gremlin, SparkQuell, making it easy to query and analyze graph data at scale. The service offers high availability, durability, and security features, making it suitable for a wide range of web-based applications, such as social networking, fraud detection, or in this case, the relationship between the terms in the semantics of the articles. The last service that we are using is the Amazon Web Services 3. That is the simple storage services from Amazon Web Service. That is an object storage service that offers scalable, durable, and security storage for any type of data. S3 is designed for durability, ensuring that data remains available and intact even in the event of hardware failures or natural disasters. The service offers features such as versioning, encryption, and lifecycle management, making it suitable for a wide range of use cases, including data backup, archival, and content distribution. We will talk later about the data lake concept that is based basically in these services. So this is a diagram of the platform where we can see like the main pieces, starting for the front end, and then that is the part with the part that the user interact with. And this is talking with the orchestration service that orchestrate the other services, such as the machine learning models or the information that is safe in the data lakes. And finally, we will find the databases where the information is properly safe for faster and better consulting. The application, as we said, we have the user interface. Also, Xavi So where the user can interact with the different services and can load the data that it will be saved in the S3 model. The orchestration layer is very important because it automates the workflows, ensuring tasks that are executed efficiently and resources are allocated optimally. Automation streamlines process while efficient resources management minimizing costs. And this also allows us to use the platform on demand, executing and the executor ensures that tasks are complete promptly as the user needs. So as I say, we want to talk about the concept of data lake. This is the layer where we can save all different kind of data. The users maybe want to load data in a CSV format, but then the responses of the model can be in another format, like for example JSON. So this is the layer that allow us to save all types of data, have all the data centralized and accessible from the front end and from the other users. And finally, we have the different databases that we are using in the platform. We have two types of databases. Basically, we have the SQL database, that it would be like the more traditional type of database that is for structured and organized systems. It allows us to save a large amount of data and facilitates the management, access, and update of the data. While on the other hand, we have also a graph-oriented database that, as I say, is based in Neptune and is for represent and understand better relationship between entities that for this particular project it would be needed. And with these services, we can complex and highly interconnect data in a best way. But that was a little fast. But if anyone have any question about the platform or something that we want to discuss.
Thank you, Rodrigo. Yeah, we'll do it at the end of the session. Okay, we are collecting some questions. So let's move to the next speaker. We are very happy to have here Shyam Gopalakrishnan. He is the coordinator of the FindingPheno project, which is a sister project from GLOMICAVE. So happy to share here experiences and presentation. So Shyam, he is associate professor at the University of Copenhagen. He has been working on developing and applying computational solutions to biological problems. And his research currently is focused on developing methods for multi-omics integrations with a focus on host microbiome interactions, both at short and long evolutionary timescales. So thank you, Shyam.
Thank you, Biotza. Can everybody hear me well? Super. So first, I'd like to thank GLOMICAVE for putting on this lovely conference at the end of their project. And I'm happy to see all the work that's been done here. It kind of reflects the challenges and opportunities that we faced as well. So I'm very happy to have this opportunity to talk about FindingPheno. As was mentioned already, FindingPheno is GLOMICAVE's sister project. We are also in the business of figuring out ways in which we can develop better methods to integrate multi-omics data. And in our case, we are also focused on very much on this idea of microbiomes playing a pivotal role, and therefore looking to see how we can integrate microbiome measurements with host biological processes. I want to give we are not close to finishing yet. We are in our third year, and so we have another year and then some left. And so we are not going to be nearly as well polished as some of the other GLOMICAVE presentations, which I got a little bit jealous of. but hopefully I can give you a broad overview of what FindingPheno is and some of the advances we've made and some of the resources that are already available for the general public to use. With that, I'll get started. Before I get started, I do want to say FindingPheno obviously is an EU-funded project, and we are made up of 8 different partners, 5 research institutions, coordinated here by University of Copenhagen. I'm the coordinator for the project. And we have 3 industrial partners as well, QIAGEN, Christian Hansen, and New York Bio. And I'll mention them as we go along when different roles become obvious. Before I get started, I am at the institution of a university, so I will be a little bit pedagogical in my presentation. Pardon if you know already all of this, and just an introduction. So first, let's look at what the classical model for phenotype evolution looks like. The classic model for phenotype evolution is the idea that phenotypes are shaped by genotype and its interaction with the environment. Either they can independently affect the phenotype or interact in some sense. And we have various tools already to study different parts of these interactions or these different data sets themselves. We all know GWAS, we all know selection studies, population genomics. So we know the different pieces how to study them. But integrating all of this has been quite a challenge that's been around for quite a while. It's not a new problem. But there's a new player on stage now as well. One principal component that's been missing, and over the last decade that's been fairly well understood to play a very important role in phenotype evolution and phenotype changes, is the microbiome. When I say microbiome, I don't just mean microbiome in your gut, although primarily in this project, we're going to be looking at gut microbiome, but any kind of microbiome, skin, gut, sex organs, whatever it may be. And so we need to update our model for what phenotypic evolution looks like. So our phenotypic evolution model should now look like this, where we have environmental factors interacting with both the host genetic and microbiome factors together into changing some of these processes that occur inside the host, such as transcription or metabolite consumption, and eventually leading to changes in the host phenotype, right? And so this complicated version of the reality makes it quite a bit harder for us to understand the mechanisms that might be affecting phenotypes. And so how do we study this complex biological process? Thankfully, over the last many years, there's been quite a lot of technological progress. And so this idea of using multi-omics as a way for us to understand biological process has been proposed by a lot of different people. And this is basically the idea that instead of collecting just the starting point, which is the genotypes, and the end point is the phenotypes, together with whatever environmental covariate there might be, we also can collect intermediate measurements of biological processes, such as transcription and epigenomes and microbiomes and metabolomes. This not just gives us an insight into what the state of the processes are, but also lets us answer questions about what might be the causal pathway leading from genotype to phenotypes. And I'll come back to this idea in a minute when we talk a bit more about it. So multiple omics integration, once we have all of this data, the question obviously arises, how do we put this enormous amount of data together to make a coherent understanding of the biological process, or identify parts of these processes where we might be able to intervene? So develop some interventional strategies which will let us modify the system to our liking. Multi-omics strategies, integration strategies have been thought about for a while, but they broadly fall into 3 categories. Either late integration, where we do all analysis, and then at the end, we kind of want to put the results together to understand what's going on. Or intermediate and early integration, where we take all of the raw data together, pretend like it comes from some similar tool, and try to figure out what parts of this raw data are important. Or intermediate, where it's somewhere in between. But one important bit to notice is the challenge that's faced by multi-omics integration in genetics and biology especially. And this is a problem of sample size. We would like our data set to look like panel A here, where we have a few genes and lots of donors or lots of samples. So we can learn quite well any of these nonlinear and complex structure that might be present in the data. But quite often we are in this panel B scenario, where we have lots and lots of features, which are genes or SNPs or microbiome abundances, and only a handful of samples. And so we end up this scenario where it's not very well suited out of the box to many of the machine learning techniques that are out there. So we need to do some selection and feature extraction. And this is going to be a feature of a lot of the methods that we use downstream. Now that we talked about multi-omics integration, what is the difference between FindingPheno and how traditionally things have occurred? Traditionally, we've been looking at these multiple different omics data and kind of working in silos. So the idea of late integration comes into focus here, where irrespective of whether we are working with the microbiome domain or the host domain, we take each of these omics data sets, do our analysis on it, and kind of finally aggregate the results and draw some conclusion and intervention strategies. So everything is a posteriori here. But our goal is to kind of take us out of this late integration approach and move into the early/intermediate integration approach, where we have all of these measurements and data sets from both the microbiome and the host data. We want to combine this with some evolutionary insights and incorporate some prior knowledge, where some of these ideas that Xavier mentioned about knowledge bases come into play as well, and then put that together in a joint analysis framework, which allows for multi-omics, time series, and evolution analysis to be done together. Are we there yet? Not yet, but we're getting there. And so this early or intermediate integration strategies allows a lot more flexibility and better use of this underlying information to understand how these phenotypes look. So what is FindingPheno doing about this? So we have three-pronged strategy, and it's quite in alignment with the call as it went out for the project. The first one is to come up with better methods for genotype-phenotype association, especially in the case of multi-omics. I know that I'm kind of making it very vague right now, but I'll get into details in a second. The second bit is account for heterogeneity. There's a lot of variation in both temporal and spatial aspects of the microbiome. And so we want to account for these variation rather than this classical assumption that we are measuring everything at steady state, which tends to be violated quite a bit, especially with the plastic system like the microbiome. And finally, how do we incorporate a priori information? So this way of thinking where we always look at our data set and pretend like it's the first data set ever on the problem clearly doesn't help. So how do we take this information that we already have, the abundance of it, and make our inferences while taking into account this information that's already present? So these are our three major lines of research in FindingPheno. A quick sidebar before I get too carried away within FindingPheno is that all of this process of method development, identification, validation, and testing would never work without access to some world-class data. And luckily for us, we have access to data from a EU-funded project, which finished a couple of years ago, called HoloFood, which was looking at production systems in salmon and chicken and looking to see how microbiomes in these animals affected their growth and affected their development in terms of their size and immunity. And we have access to this data, and luckily for us, they have the same individuals who've been measured for multiple different omics analyses. So it's not as if we have a scatter shot of different types from different individuals. The same individuals, for the most part, have genomics, transcriptomics, metabolomics, and microbiome-related traits as well. Now that we got it out of the way, let's look at how do we develop better integration of multi-omics data. For the most part, we look at three different approaches for multi-omics data integration. The first one is this idea that how do we go from correlation to causation? We look at mostly causal models. And in this particular case, we are looking at either conditional independence or independent ancestry associations to see if we can take that arrow which tells us about correlation and convert it to a causal effect. So we can go ahead and then say, oh, you know what, this is causal, therefore we can go ahead and do something about it and change it the way we want the outcome to look. We also use Bayesian hierarchical models. These have been used quite a lot for a while on different multi-omics data sets. And the idea here is understanding some level of latent factors that might be present and using those latent factors as the bits that we want to really work with. Finally, we use a reasonable array of machine learning methods, including sparse regression, conditional autoencoders, and we also lately have been using quite a bit of graph neural networks as well, which have worked very well. And as of right now, we are still in the process of improving them. But at first glance, they seem to be working quite well. Much of this effort is led through researchers both at the University of Copenhagen and at the Shyam Gopalakrishnan Foundation in Lisbon. So let's look at a quick application of some of these methods to salmon data. And we are here going to look at the causal models looking at salmon data. The salmon data contains transcription and transcriptome and microbiome from about 120 salmon with 2 different time points. And there are 2 different treatments, so 2 different feed types. And our goal is to be able to predict what the size of the salmon is. And eventually, obviously, we want to understand what are the biological variants that are affecting the size of the salmon. When we do this, this work is led by two young researchers from Champalimaud Foundation. When we take our 20,000 different observations, our features, and reduce it down to a manageable set of about 300, and we look at independence of these causal variables on our data set, we find that there are a few genes that pop up. But if you notice, there's an abundance of hemoglobin that shows up every time when we look at any of these measurements, which seem to be affecting or be affected by weight. And when we check for this, we find that this is very significant, even when we control for arbitrary correlations that might pop up in the genome. And to our surprise, actually not to my surprise, a lot of the people already knew this, hemoglobin has been implicated as a mechanism which gets affected quite a bit by body mass. What was strange was this data set's gone through all of the traditional testing in hollow food, but hemoglobin never came up. It was only when we did this causal prediction based on accounting for the correlation structure of the data that we start to see it crop up. So it's quite reassuring to see that some of this finding is not particularly novel. The second prong of FindingPheno looks at heterogeneity in microbiomes and looks at how this heterogeneity can be handled, both on the temporal and the spatial axes. And this work is primarily led by investigators at the University of Turku and the Center for Ecological Research in Hungary. And some of the classical models, including Lotka-Volterra models, which look at predator-prey interactions, and common resource, consumer resource models are used for modeling the dynamics of these systems. So again, as an example, here we are looking at the dysbiosis in the salmon gut, and this is from a different study. It's not from hollow food. Dysbiosis in the salmon gut, where let's first look at the left panel, where we're looking at healthy and sick fish and their gut content microbiome. Here, the yellow is mycoplasma and the blue is Allivibrio, which is a kind of a indicator of pathogenic bacteria in the gut of salmon. And we can see here that for the healthy fish, it's mostly dominated by mycoplasma. For the sick fish, it's mostly dominated by Allivibrio. But after treatment, something really goes wrong. Everybody seems to be quite dominated by Allivibrio, both in the gut content and the gut mucosa. And so we started modeling this, and through simple interaction and consumer resource models, which look at differential consumption of these resources, we were able to model the interaction between the immune system and components of this gut, which give rise to this bistable system where you either get dominated by Allivibrio or by mycoplasma. Again, this we can take back to our collaborators who work on salmon and get it validated and make sure this is the way for us to control some of these infections. I forgot to tell you, this infection for the sick fish is not a gut infection. It's actually a parasitic infection of the skin. So clearly the change in the gut microbiome is primarily driven through immune system response. Final prong of FindingPheno involves looking at a priori knowledge. And here we've been using some of these databases that are publicly available on gene-gene interactions and gene metabolite interactions to look to see if we can use that as the backbone for some of the observations that we see and figuring out which microbe or which gene might be causal for the phenotypes that we care about. I'm mostly talking about phenotypes here from the perspective of production animals, but clearly this is widely applicable as well. And here our primary focus has been graph neural networks until now. This is not from us. This is from a paper that we are inspired by, but we are here looking at 2 different ways of representing the data, either directly as gene-gene interaction networks that we can download and use from databases like KEGG, or as a graph neural network where genetics determines the connectivity of the network and each node is a sample. And both of these approaches until now have been extremely promising. So I think you will hear more from us on this over the course of the next year. Final part of our project is also leveraging public data sets. At the time of writing the project, this was quite an enthusiastic part of me was quite enthusiastic about this. But looking at the state of public databases for multi-omics studies, now I regret having put this in the database. But we're looking at tomatoes, soybeans, honeybees, maize, and barley. We have about 100 different studies from ENA where we've already processed the metagenomics and the non-metagenomics part of the data. And we want to apply some of the methods that I've been discussing until now to these. But it comes with its own set of problems. There's a lot of missingness. There's a limited number of omics in some of these. And shockingly, there is absolutely really, really poor metadata annotation for some of these studies. I know that all of you listening are very good at submitting your data and metadata, but please do pay attention to how good the quality of the data that you submit is. Because even though we have a lot of data, much of it is quite difficult to use directly, given the state that we are in right now. And I would just want to finish off with like a couple of things that I want to note is FindingPheno's methods are available for use already. Many of them are mature, some of them are on their way to maturity, and they're being deployed either as standalone R packages or online tools that you can directly use, or as standalone tools that you can download to your computer and install on your own machine. There are simulation frameworks, and there is also direct integration to EBI's platforms like Magnify and ENA. So you can download the data and process it without having to leave, doing the whole thing programmatically without having to go back to your command line. And so we are quite excited about some of these packages that are coming out. The work is primarily led by Rob Finn's team at EBI and Leo's team at Turku. There are also these online tools, like I said. So if you go to Magnify, you can find FindingPheno. You can find some of the assemblies that have been done based on publicly available data and the catalog of public data sets that have been used. And all of these are programmatically available. So you can go in there, you can open up a R or a Jupyter notebook with R or Python, whatever you're most comfortable with, and play around with the data as they've been processed. I told you already, they've been processed. So all of these data sets are available as summaries. And finally, there is a plethora of tools available for both understanding the microbiome analyses that we've been developing methods for. So there's a whole book on it led by Leo. It's called Orchestrating Microbiome Analysis. It's freely available online. So, and with example data sets, so you can play around with it. It's part of the Meoverse. And HoloFood again, is a data portal, which is now being programmatically provided access through based on some of the work that's been done in Magnify and FindingPheno. And with that, I just want to say thanks to everybody again for listening. And if you feel free to get in touch with me, either on any of the social media platforms of FindingPheno or directly through my email, I'd be happy to chat about any of these if you're interested in any of our methods. Thanks, everybody.
Thank you. Thank you, Shyam. I know we are a little bit in delay, but just wanted to at least pose one question to each of you. And maybe I would start with Xavier. You've been talking about how to use GLOMICAVE, but maybe one question here is it free of charge? How are we dealing with this idea of keep on improving the tool, but then as well, making openly available.
Right. Yeah. So it's so our idea is to make it free of charge, at least for some parts, because of course we want the people to be able to use it, especially the non-commercial users or users that don't want to use it to exploit it commercially, so for scientific purposes. But the problem is that, of course, maintaining such a large database, maintaining such an important resource takes, it needs a lot of money. You need really to pay the people that takes care of and maintains this platform. So we're thinking of going with a hybrid type of scheme where you will be able to use the platform for non-commercial purposes, and then people that wants to do it or to exploit commercially the features of the database, or users that really want to use a lot of resources from the platform, they will have to contribute to the maintenance of the platform in an economic manner.
Okay, thank you, Rodrigo. for the additional explanation. And then, Shyam, we also have a couple of questions for you. One, it says, how to use methylation data in these omic analysis. And then another one, it refers to if the metaproteomics data is generated within, or it is foreseen within FindingPheno to get info from the microbiome side, or if it is assumed to be used or to be extracted, let's see, from the metatranscriptomic data.
Yeah, I can answer both of them. Let me begin with the epigenetics question. Epigenetics is just like mostly variation that we see, so genomic variation, SNPs. So if you care about whether, for the most part, where we find interest in epigenetics, it's its effect on the expression status of many of these genes, and in which case it's kind of a nuisance parameter for the most part, because you have directly to measure the expression, so you don't really need epigenetics. But if you do need epigenetics, if you can characterize them either as a continuous variable on the amount of methylation at any given CpG site, or if you can dichotomize it to absence or presence across samples across these methylation sites, it's just a matrix at the end of the day. So we can, all of these measurements can be used just as any other measurements in terms, but most similarly, it looks like the genomic variation at SNPs. Now, let me get to the second question. The idea of metaproteome is absolutely correct. We do not actually have direct measurements of metaproteome. We do generate these just from the metatranscriptomic data. And we do have measurements of the metabolome, though. So we can actually figure out what the metabolome looks like. But for metaproteomics, the question is absolutely spot on. We primarily depend on the metatranscriptomic data to figure out what compounds are going to be present.
Currently, still FindingPheno, and still and will run for 6 months or still 1 year?
No, another year.
Another year, full year. Okay,
Yeah,
so
yes.
yeah, we will be keeping following also your results.
Thank you.
So yeah, we closed let's see this session second technical sessions. Thank you very much, Xavier, Rodrigo, and Shyam for your presentations. And we'll just jump to our let's see roundtable with the use cases leaders, to have more chat-to-chat discussion on the GLOMICAVE, the experience, and the results within the different sectors. So yeah, let's move to next session. Thank you very much.